Yes, children, I hope the screen is visible. Just raise your hand if you can see the screen. Okay. And also one more thing, can you see the pen moving on the screen?
Yes, ma'am.
Yeah.
Okay. Children, I think I'll have to switch off my video because it's lagging. No, I think now it's fine. Okay. You all can switch on your videos. Right. So basically, we have to start with the Mendel's experiments. Okay. What experiments did he do and the conclusions he came with? So we have to basically talk about the inheritance of one gene. We are talking about the experiments done by Mendel. Okay. So out of it, the first one is inheritance of one gene. Right now, don't write anything from this slide. I will tell you what to write. Okay. So inheritance of one gene can be explained by monohybrid cross. Mono means one. That means Mendel conducted an experiment to study the inheritance of one character. And what was that one character? That one character was height of the plant. Okay, so basically he conducted the experiments on pea plants. So he was actually seeing the inheritance pattern of the pea plant in terms of its height. So only one character is there, that is height. That is why the cross or the experiment he performed, the name of the cross was monohybrid cross. Why mono? Because only one character inheritance he was studying, and that was height, right? So the basic outline of his experiment was, what he used to do, he used to make a cross between the plants. Okay, when the plants were crossed with each other, the new generation came up. He used to study that new generation, and on the basis of that, he used to give conclusions. That's the basic outline of his experiment. He used to make the plants cross with each other to study the inheritance pattern in subsequent generations, right? So when he said that the parents that we are crossing, the parents that are getting the plants which are getting crossed with each other, he referred to them as parents. And the new generation that came up, he gave it a name as F1 generation. Further generation which came up, he said that it is the F2 generation. Okay, so this is the basic outline of his experiment. Now we have to talk about the monohybrid cross. So first of all, you should know what is a monohybrid cross. Okay, what is the definition of a monohybrid cross? Is the cross between two parents differing in one pair of contrasting character. One pair of contrasting character means, one pair means that is height, which Mendel took. Okay, we are talking about the Mendel's experiment, the cross between two parent plants which differ in one pair of contrasting character. That is known as the monohybrid cross. So we have to see the monohybrid cross of Mendel. Okay, Mendel's experiment. So you see this. Children, let me tell you one thing. When we are studying crosses, the way you have to make the cross, the way you have to draw is very important. Everything you are writing while making a cross counts, and everything has marks. So while making this cross, while making the monohybrid or the dihybrid cross, be very particular of what all you are writing. Because if you will miss anything, if you will make an incomplete cross, your marks in the exam would be deducted, right? So I will teach you how to draw, I will teach you how to make it. We are studying about the monohybrid cross first, in which Mendel considered only one type of character in the plant, in the pea plant, that is height. He was actually trying to see the inheritance pattern of the pea plants in terms of their height. So how it happened? First of all, this is one thing. Let me come to this. First, I will explain you with this. Okay. And you will draw this. So see, just a second. Yes. So it's a monohybrid cross. Okay. Cross between two plants. One plant was tall in height. Other plant was dwarf in height or short in height. Okay. So when I say that the plant was tall, that means I am referring to the phenotype of the plant. In the previous class, I told you phenotype means the observable character or the physical appearance of an organism. So phenotype of the plant, one plant is tall, other plant is short. Okay. And P for parent. These are the parent plants. Okay. So phenotype is tall and dwarf, two plants. The genotype of that particular plant is capital T, capital T. So basically there is a rule in genetics. Okay, the rule is that for the dominant trait, you have to always use a capital letter. Okay, and for a recessive trait, I will tell you what is a dominant trait and what is a recessive trait, although I have told you in the previous class. So it is a rule that you have to always denote a dominant character with capital T, whereas a recessive character with small letter. So when the plant is tall in height, the genotype of that particular plant is capital T, capital T. When the plant is short in height, the genotype of that particular plant is small t, small t. Okay, come to this previous figure once again. See this. Okay, so parent, this is the parent plant. Okay, one with capital T genotype and one with small t genotype. Okay, then we have to take out the gametes from these plants because the crossing will take place. I am giving you one advice that whenever you will write gametes, please circle them. Like for example, this is a plant with genotype capital T, capital T. Okay, you are taking out a gamete. So one T will come for gamete formation. So you circle it. You circle the gamete like this. Okay. Then see the other plant that is short in height with a genotype small t, small t. Take out the gamete. You will take it one, small t, again circle it. Okay. Now what will happen? You are going to, like these gametes will fuse with each other. Okay. So how is it happening now? See this. Gamete has come. From here, it is capital T. From here, it is small t. Right. So what the F1 generation would be? F1 generation would be all plants like capital T, small t. Okay. That means T is the dominant trait because it expresses itself. That means it's a dominant trait. So that means the plants would be tall in height because it has one what? Dominant allele present in it. Again, I'm writing it. See to it once again. I'll just erase everything. So, one parent plant tall in height with genotype capital T. One parent plant short in height with genotype small t. Then gamete. Here in this picture, gamete formation is not there. So now I have made it, I have written it with a pen. You can also write it. So the gamete here would be capital T circle. Here would be small t circle. And then when they are crossed, you get the generation of plant, next generation of plant with genotype capital T, small t. Okay. Now, because it has got capital T in it, that means it is actually the dominant one, dominant allele, and it is denote dominant for what? Tall. That means all the plants, the height of the plants they are tall. Okay. Now, what is this written? Homozygous dominant, homozygous recessive. I told you when the alleles, they are like same, capital T, that is the homozygous condition. Whereas here, when the alleles are different, capital T and small t, this condition is heterozygous. Right? Then what you did? No, not you, Mendel. What Mendel did? Mendel made a cross between the F1 plants. What is the F1 plant? F1 plant is capital T, small t. Now, what he did to obtain the F2 generation, he made like the self-pollination happened between the F1 plant. So F1 plant was capital T, small t. He crossed it with same. Like the F1 generation plants, whatever the plants were there, he made the plants self-pollinate. Okay. So capital T and capital T, same generation of plants. Then again, what he did, as you can see here, how the cross happened. What will be the gametes? Sorry, the spelling of gametes here is wrong. After T, E will come. I am talking about the F1 generation. I'll just erase, sorry, F2 generation. So F1 generation, the F1 generation which we have received is capital T, small t. This is the F1 generation. Okay. Now they are crossed with the same generation. Okay. Now gamete formation will take place. So let's take out the gamete. Capital T, it will give one gamete, capital T, it will give small t. It will also give capital T, it will also give small t. Now we have to write the gametes. How do we write the gametes? This is the formation, Punnett square. It is known as Punnett square, in which you actually calculate the probability of plants. So this is the F2 generation I'm calculating. How I'm going to write it? 1 second. F1 is capital T, which will become the parent for the second generation. Okay. So gamete from here, this, from here, this. Now I want to make the F2 generation. How I am going to make? I will make a Punnett square like this. Okay. I will write the gametes. Male gamete, we always write here, and female gamete, we write it here. I have written the symbols. So the gametes are same, capital T here, small t here, capital T here, small t here. You just have to multiply now. First of all, this capital T multiplication will happen with these two. Then this small t multiplication will happen with these two. So when I multiply capital T with this capital T, I will get capital T, capital T. When I will multiply the same capital T with this small t, I will get capital T, small t. When I will multiply this with this, again I will get capital T, small t. And when I multiply this small t with this small t, I will get small t, small t. Okay. If I write the phenotype, what is the phenotype? Capital T, meaning the plant is tall. Capital T, small t, again the plant will be tall because we know T is the dominant character which is able to express itself, and small t is the recessive character which is not able to express itself in the generation. Then we have capital T, small t, again the phenotype would be tall. Now there is small t, that means one plant is short. So what he showed you from monohybrid cross, he showed that when he was trying to obtain the F1 generation, he saw that all the plants were tall in height. Okay. And when he came to F2 generation, he saw that one particular plant was short in height. So Mendel said, as per Mendel, he says that one character, that is the dwarf, the character, short character for short height, the character for short height, which was not able to express itself in the F1 generation, it later got expressed in F2 generation. That means in F1 generation, this trait for short height was hidden. That is why it is recessive, because it was not able to express itself.
So it's said because it's more of a probability for tall height plants.
Okay. So this is the monohybrid cross by Mendel. And based upon this monohybrid cross, he gave law of dominance. Okay. I'm quickly giving you the recap of this slide, and then you have to draw this in your notebook right now. Okay. And then I will tell you the law of dominance based upon this monohybrid cross. How you have to make it, don't get confused, keep it simple, because the way you draw is really important. How you have to draw it? Just give the heading monohybrid cross, right? Write like this: parent, write this phenotype, genotype, tall, dwarf, all these things write down. In between, you need to just add gametes here, because in this diagram, gamete is not written. Okay, so just write gamete, and here, this is the gamete, capital T circle, and here, this is the gamete, small t circle. Okay, and then you can show it like this, that you are doing this. So the F1, in the F1 generation, you will get the plants with the genotype capital T, small t, which is all tall, and they are heterozygous. Now you have to obtain the F2 generation. So you will self-pollinate these plants. Self-pollinate as in, you are going to just capital T, small t will be crossed with capital T, small t. Same way. Same way when here it is written, na, parents, this thing. Same way you have to write for F2. Here they will be parents. Parents for second generation. Capital T, small t, capital T, small t, and take out the gametes. Gametes. This is one gamete. This is one gamete. I hope I'm not confusing you all. This. Just take out the gametes and then make this F2. Draw this Punnett square right here. And yes, one more thing. Do add, make a symbol that male gamete is here and female gamete is here. Okay. Are you clear what you have to draw? Everybody. If you will say, I will draw it on a blank slide. If it's not clear from here, please let me know, children. Is it clear to everybody, like what you have to draw? What you
Can you
have to
draw it on a
write?
blank slide, please?
Huh?
Can you draw it on a blank slide, please?
Sure, sure. Okay. 1 second. Mm, just give me a second, huh? For monohybrid cross, I'm making the cross. Like I'm telling you how to draw it. For dihybrid, you are going to make a cross. Okay? Just a second. Shivansh, are you listening to some music?
No earphones, just humming.
Okay. 1 second. Yeah. Please tell me if you all can see the blank slide.
Yes, sir.
Huh. Okay. Right. So monohybrid cross, the heading you can give on your own. So parent. Okay. So parent phenotype. One is tall. One is dwarf. Then parent genotype. Parent genotype. Capital T, capital T. Small t, small t. It is known as homozygous dominant because both the alleles are in capitals. Dominant, sorry, homozygous dominant. Then this is, the condition is, het, sorry. Oh. This is also homozygous. I'm sorry, I was writing heterozygous. This is also homozygous dominant condition. Then you have the gametes. So for this, the gamete will be in circle. For this, the gamete will be this. Then you are obtaining the F1 generation. What will be the F1 generation plants? How will they look like? So all the F1 generation plants would be capital T, small t, and this condition is heterozygous. Heterozygous because it has one capital also and one small also. And if I talk about the phenotype, it is tall. All plants tall in F1 generation. Now we have to talk about the F2 generation. So this F1 generation plants will become parents for the next generation. So I'm writing this F1 plant like this because now the cross would be happening between the F1 generation plants. So again, the same thing. You are writing these are the parents. These are the parents. Okay. It's your choice if you want to write the genotype and phenotype again. Then I'm talking about the gametes. So the gametes, this will give this will give this will give this, and this will give this. Then I'm obtaining the F2 generation. So for this, we will be making a Punnett square is a graphical, is a representation basically to calculate the probability. So this is how you make it. One second. This is how you will make a Punnett square, right? And you will be writing like this. The gametes, the male should be written here. The female should be written here. Then write the gamete. One is this. One is this. The female is also same. The gametes, this. Now start multiplying it. Capital T, capital T, capital T, small t, capital T, small t, and small t, small t. You can write the phenotype in the Punnett square only. Like capital T, they are tall plants. So write tall here only. This will be also a tall plant, right? Tall. This will be also a tall plant because it has got capital T in it. This is small t, which means now the plant is dwarf in height. Okay, this you have obtained the F2 generation. One question which comes is that what is the genotypic ratio and phenotypic ratio? So for monohybrid cross, I am telling you the genotypic. For phenotypic, for dihybrid, you are going to tell me. So genotypic ratio means, children, see the genetic constitution, genotypic ratio. As in, I'll use another pen for this. One second to highlight it. I'm using blue. Yeah. So see, capital T, capital T. In this Punnett square, is there any other capital T, capital T? Please tell me this.
No, ma'am.
So for capital T, I have written 1 because I find it only 1 in the box. Now capital T, small t, how many are there in the box? Two. Capital T, small t in two boxes. One is here, other is here. So 2. Small t, how many? Only 1. So the genotypic ratio comes out to be 1:2:1 for monohybrid cross. What about phenotypic? See the physical appearance of the plant. How many plants are tall in this Punnett square?
Mom, two.
Tall
Sorry,
plants.
three.
Very good, Jivtesh. 3. And how many short plants?
One.
So the phenotypic for monohybrid crosses 3:1. This so you have to learn. Please learn this, okay? The ratios, learn it. That this is the ratio for monohybrid, this is the ratio, this is the genotypic ratio for monohybrid, this is the phenotypic ratio for monohybrid. Learn it, okay? Because based upon these ratios, you will be getting some mathematical questions in the exam, okay? So in the sample paper, there is a question. So don't worry, we'll discuss that. That is why I am saying learn the ratios. And be very specific in making the crosses, please. अपने आप कुछ भी कहीं पे भी नहीं लिखना है. जैसे रिप्रेजेंटेशन है प्रॉपर, बहुत नीट और अच्छे से इसको ड्रॉ करना है. ओके?
Yes, ma'am.
Because it has marks. The way you represent it, you're like according to that only you will be awarded marks. So be very particular in making the cross.
Ma'am, so for after generation, what will be like the offspring? It'll be tall, right? Capital T, small t.
F2?
Yeah,
See, you see the Punnett square, what you can see.
it should be tall because majority is tall only.
No. They are, majority of them are tall, but the one plant here is short heighted because it has got both small t. That is why it is able to express itself this time, because both the alleles, they are in recessive condition. Okay. If it was like capital T small t, then the plant would have been tall. It cannot be short because a dominant allele is present with it. And when a dominant allele is present with it, obviously the dominant one will show its action. But when both the alleles, they are in small, they are small t, then only that character will be shown, and that is that is dwarf height. Majority of them are tall, but out of them, one plant has shown the recessive trait which was hidden in the previous generation.
So we'll take the hidden trait, right? The recessive trait.
For
Like for the outcome of this one F2 generation.
no. Outcome of F2 generation will be this only. 3:1. That means three
Oh,
plants tall, one plant short heighted. For F1
okay.
generation, all the plants tall. So the outcome is this only. Have you drawn this, everybody? Children, I cannot hear you. Can you hear me? Okay. Jivtesh, have you drawn it? Very
Yes,
good.
ma'am.
Very good. How many of you are still drawing?
I've drawn it.
Very
No.
good. Yes, Shivansh?
Did.
Okay, who is left? Is there anyone who is still drawing? Because then I will wait, otherwise I will erase it. Nobody? Sure. So that means I should erase it. Please stop me if you haven't, okay? I'm erasing it. Yeah, so basically from this monohybrid cross, a law came up. Okay, that is law of dominance. Those students I think Shivansh and Dhruvi, in your school, this chapter has been over. So who will tell me the law of dominance? Yes, Shivansh?
Basically, a law of dominance is this. We have two traits. One is dominant, one is recessive. The dominant trait is always going to excel. Let's say you've got a genotype in which the dominant trait and the recessive trait both coexist. The dominant trait is going to, obviously, as it's in the name, the dominant trait dominates the recessive trait, and the dominant trait gets expressed in the organism. So, for example, if I were to give capital T and small t, the genes for height, capital T would express the trait of being tall. Lower T would express the trait of being short. Assuming
Hmm.
that capital T is the dominant trait,
Hmm.
that would mean that capital T would express itself and lowercase t would be suppressed.
Right. So what is the law?
Mom, that's a law. You've got two traits. The
No, see.
dominant trait is going to express the trait, and
Hmm.
the recessive trait is going to be suppressed by the dominant trait.
Exactly. So whenever you are telling the law, right? So you have to like state the law as it is. You cannot, tell the law in your own words, right, Shivansh? So you just have to be like the whatever you have explained, that is perfect. Okay, your explanation is fine, but state the law.
we haven't learned the exact wording,
Oh,
but I
fine.
can tell you this.
Okay, fine. I'm telling you. Just a second. See this. See this. Law of dominance. Can you see it? I will explain you. Your explanation was perfect. Okay. But when in exam, you are asked to write a law. Okay. Because we cannot write the laws in our own words. Okay. So laws have to be written as it is. Like whatever the law Mendel gave, that was the law of dominance. These are the pointers which were given by Mendel. Okay, so we have to write them as it is. What does the law of dominance states? It states that the characters are controlled by discrete units called factors. What is factor? You know, Mendel at that time, he did not know about the term gene. Gene term was, the term came up very late. Okay. So instead of gene, he used to use the term factors. Okay. So he says what? That characters are controlled by discrete units called factors. Right. We know it. Whatever the traits we have that are controlled by gene only, right? So this is the first point of the law of dominance, right? The second point of the same law states that factors occur in pair. We know that the genes, they occur in pair, right? The third point of the law of dominance states that in a dissimilar pair of factor, dissimilar means I'm giving you the example of dissimilar means capital T, small t. They are dissimilar, right? One is capital, one is small. Okay. So the dissimilar pair of factor, one member of the pair will dominate the other. That means in this condition, capital T, small t condition, who is going to dominate? Capital T is going to dominate the small t. These are the 3 pointers of the law of dominance. Everybody, please write it down as it is. When you are asked to state a law, okay, don't modify it, don't change it, okay, because these are the direct statements given by Mendel and it's a law. Okay, so we cannot, we cannot add our sentences to it. Just state them directly. You have to learn it as it is. Okay, if you are asked to explain the law, then Shivansh, whatever you explained to me just now, you will write that also. Okay, if you are asked to explain. But if you are asked to state the law, then you will write these 3 points. I hope I'm making it clear, everybody. Say yes, na?
Yes, ma'am.
Yes, ma'am.
Okay. When you don't say yes, na, you all demotivate me.
Is there any law of recessive or only dominance?
No, recessive thing is itself getting explained by this law only. है ना? इसी में explanation आ रहा है उसका भी. ऐसे
Yes.
separate law कुछ नहीं दिया था. इसी में you are getting to know what it means, right?
Yes.
So quickly write down and let me know once you are done. Do let me know because I'm waiting for all of you. Oh, I'm sorry, I changed the slide by mistake. 1 second. 1 second. Yeah. Written, children.
Done now.
Everybody. Shall I move to the next slide?
Yes, ma'am.
Okay. Now we have studied the first law given by Mendel. Um, okay, we studied the monohybrid cross. Now you all know how to draw the monohybrid cross and what all things you have to write and include in that cross. You all know this. Okay, and based upon this monohybrid cross, you are now clear with the law, first law of inheritance by Mendel, that is law of dominance. Now let's move to the another type of cross. This was monohybrid cross. Now we will be talking about the dihybrid cross. Okay. Now, okay. Because I have told you the definition of monohybrid cross, who will tell me the definition of dihybrid?
Ma'am, so in the dihybrid, like definition, exact definition, I don't remember. But
It's
then,
okay.
like dihybrid crossing means that Mendel took one green and yellow color pea seeds.
Hmm.
Then he took the gametes, like the gametes fused together, and then there were variety of seeds, round and green, round and yellow, then the wrinkled and green, wrinkled and yellow. So he used all of them separately to get different results, and every time he used different categories, a new type of like seed came out. Like if he
Okay.
used red and yellow, then sorry, round and yellow, then wrinkled and yellow came out.
Exactly. Okay. So you just gave the background. It's okay. It's good. Okay. Fine. Fine, Jiftesh. See, dihybrid cross. I told you the monohybrid. Monohybrid means mono means one. That means the cross which was actually used to study only one type of character inheritance. That was height. Here in dihybrid, Mendel was trying to study the inheritance pattern of two characters. Di means two, di means two. So that is why he took two characters of the pea plant to study. What were those two characters? That was the seed shape and seed color. These two traits he used to study the inheritance pattern through a dihybrid cross. In the monohybrid cross, it was only plant height. Here it is the seed shape, one trait, and the other trait, seed color. Right? Again, the same thing. You know how to write. Dihybrid cross, you are writing the parents. Write the genotype and the phenotype of the parent. Phenotype is the observable characteristic, how the seed looks like, the seed that he took. The phenotype of that seed that he considered as a parent seed was round in shape and yellow in color. One seed. Second seed that he took as a parent was the seed was wrinkled in shape and green in color. Write the genotype. For writing up the genotypes, how he used, what he used to denote. He denoted round with capital R, okay, and he denoted yellow with capital Y, okay. Now he cannot change the letters. You will never change the letter. If you are using R for the shape, seed shape, round shape. So for the other shape, you have to use the same letter, but in smaller case. Don't do that for wrinkled, you will write W. No. If for a shape you have used a particular letter, if you have used it, if Mendel has used this capital R for round, then for wrinkled, he is going to write it in the recessive state, that is small r, small r. For yellow, capital Y is there. So for green, it would be small y, small y. Now, gametes. Circle them, okay? From 2 Rs, 1 R will come. From 2 Ys, 1 Y will come. From 2 small r's, 1 small r will come. For 2 small y's, 1 small y will come. Circle them. Okay? Circle the gametes always. And then what you do? Cross. What he obtained in the F1 generation? All the seeds were round in shape and yellow in color. Why? Why round in shape? Because it had one capital R. We know the law of dominance. Why yellow? Because it had capital Y. We know the law of dominance. So all the seeds in F1 generation were round and yellow in color. Round in shape, yellow in color. This is the story of F1 generation. I'm repeating again. Parent genotype is capital R, capital R, capital Y, capital Y. Because now there are two traits, that is seed shape and seed color. Phenotype is round yellow. For other parent, the phenotype is wrinkle green. The genotype you are writing as small r, small r, small y, small y. Gamete, take 1 from both. Capital R will give 1 R. Capital Y will give 1 Y for gamete formation. Small r will give small r, 1 small y will give 1 small y. Cross them, you will get the F1 generation as round yellow. Draw till here. Then I will move ahead. Okay. I hope you all have written. Yes or no?
Yes.
Okay. Now we will be moving to the F2 generation. Okay. Now this F1 generation will become the parent for the second generation. Okay, this F1 generation will become the parent for the next generation. So cross, again self-pollination with these F2, F1 generation seeds. So again the same thing. Cross will be made between the same seeds of the F1 generation. So these have become the parent for the next generation. You will write parent here. Okay. Then next thing that we write is gametes. We write is gametes. How are you going to make the gametes now in this case? Just take this one parent because it is the same. So the gametes would be same for the second parent also, na? Take out this capital R, capital Y, small y. How will you take the gamete? Capital R multiplied by capital Y is capital R, capital Y. Okay. Multiply capital R with small I also, sorry, small y also. It becomes capital R, small y. Multiply this small r with capital Y, you will get this gamete. Again, multiply this small r with small y, you will get small r, small y. And same with this second parent also. Same gametes will come for this also because they are same. So I'm not making it again. I'm just writing it. Sorry. Okay. We have got the gametes. Clear? Gamete formation clear, or should I tell again?
And can you repeat? I had like a network issue.
Okay. I'm erasing it. I'll explain you again. See quickly. This becomes the parent for the F2 generation. So cross them with same. Okay. Take out the gamete. How are you going to make the gamete? Multiply this capital R with first capital Y. Then multiply this capital R with small y. Then multiply this small r with capital Y. Then multiply this small r with small y. And it would be same for second parent also. So I'm writing it and like this only. I'm not making it again because you know how it came. Clear?
Okay, ma'am.
Okay. Now these are the gametes for F2 generation. Clear? Now time to make a Punnett square because we have to calculate the F2 generation seeds. How are you going to do this? I'm coming to a blank slide. Okay. Oh, where is my blank slide? Yeah, here it is. I'm erasing this. Time to make a Punnett square. I hope you have written that parent and gamete for dihybrid, or should I write again?
You can continue with the Punnett square.
Okay. So now it's time to make the, there were 4 gametes with each parent, with male and the female. So 1, 2, 3, 4. 1, 2, 3, 4. The gametes were capital R, capital Y. Come on, tell me the gametes. You will tell me. What were the gametes? 4 gametes. Tell me.
Ma'am, small r, small y.
Tell me in the sequence.
Okay.
Yes.
Uh, small r, capital Y.
Okay.
Then small r, small y.
Sure.
Mm,
See this. वैसे तो it doesn't make any difference, but we should go with the Mendel's work only the way he did it. Okay. These are the gametes. Na?
yes.
Hmm. Capital R, small y. Okay. Capital R, small y, small r, capital Y, small r, small y. And the same for the male parent also. Same gametes. So this becomes this becomes this becomes this. Now, you will help me fill this Punnett square.
Yes.
Okay. So, whoever wants to volunteer, come on, tell me, what should I write in the first box?
First box it will be RR. Like both the R's are capitals and both the Y's are caps.
Yes, please don't see your book or don't see somewhere else. Even if you have done it, it's a revision for you. So see this and tell me. Like, see the slide and then tell me. Fine, okay. The second box,
It'll be both capital R and a capital Y and a small y.
good. Third, quickly,
It will be capital R, small r, and both the Y's are caps.
good. Now, apart from Jivtesh and Garvit, others are also there in the class. Quickly tell me. Arey, children.
Can we fill the Punnett square in ourselves and send it on the group?
Perfect. Do that. Do that. But not only Punnett square, complete dihybrid cross. Starting from round yellow parent seeds, wrinkled green parent seeds, gamete for the first generation, F1 generation, F2 generation, then parent, then gamete, and then this Punnett square. Clear?
Yes, mom.
Or do you want me to show the previous slide where I started with dihybrid cross?
Mom, I need to check the gametes for F2. That's it.
Okay, I'm opening that, but this Punnett square, then everybody will fill on their own. Okay, so I'm showing you where I started with dihybrid. You can see it. Yeah, see this. See this. This dihybrid cross. Parents, the genotype of the phenotype of the parent, genotype of the parent, their gametes, the F1 generation, which is this, and this F1 generation becomes the parent for the second generation. Then gametes, how the way I calculated the gametes, do that, and then after that, Punnett square. And please do not see the dihybrid cross, F2 generation of dihybrid cross from anywhere else. Do it on your own. It will help you, like it's consider it as your self-assessment.
Ma'am, so from the like F1 generation parents, the RY and R capital Y small, that are the gametes, right? Like which form again?
Which I have made in a circle.
Yeah.
Yeah, these are the gametes.
No.
We need gametes, right, for the reproduction to take place. So from like what sort of gametes do these parents contributing, that I have written. Okay. Okay, I have got it from Hriday on the group. Hriday, you need to close the Punnett square, properly make it, okay? Just cross, like close it. You have just drawn the lines. You have to make it a proper Punnett square, okay? Just close it. Okay. Phenotype, genotype, gametes, F1 generation, and also write F2 generation. You have not written F2 generation. F2 generation, right? F2 generation near the Punnett square. Okay. Do this. Okay, Shivansh. Dihybrid cross, parents, gametes. Okay, Shivansh, you need to also write the phenotype of the parent. What does a parent looks like? It is a round yellow seed, okay? So you need to mention the phenotype also. Then you have F1 generation, round yellow, gametes. Okay. Again, you also have to properly close the Punnett square, Shivansh. Okay, properly draw the rows and the columns. That would be considered a complete Punnett square. Otherwise, your marks can be deducted. Hriday, you also, please, okay? Properly make the Punnett square with all the columns and okay, you have made the cross. Okay. Shivansh, in this, you have to now identify also the phenotype of all the seeds. Okay, everybody has done it. I have
Mom,
received
I'm
from
sending it. Like I'm sending it.
what?
Mom, I'm sending the pic.
Fine, fine. Till then, you all send it, and I'm filling it in my Punnett square as well, and just tally it. Okay.
Done, mom. Send.
Okay. Just quickly see this. I will make it quickly. This is capital R, small I, capital Y, small y, capital R, capital R. This, it will be this, it will be this, it will be this. Then this becomes this becomes this, and this becomes this, this, oh sorry, R R small, this and RRYY and RRYY. Done. Okay. So if you see the phenotype, this is round yellow. This is round yellow. Okay, 1, 2. This is also round yellow 3. This is also round yellow 4. Again round yellow 5. Again round yellow 6. Round yellow 7. Round green. Okay, so first I'm calculating the round yellow seeds. 7, 8, 9. Um, it's a mistake from my side. Oh yeah, I have written it capital R. Yeah. So here also I think I have done some small r. 9 should be there. I think I have written something wrong. Is it? Oh yeah, this is small y. That is why. Okay. So, oh, now it, yeah, okay, fine. So this is small y children. I thought I have written it as capital Y. Anyway, so uh, should I write it again? Or is it clear to you? Should I do it again, children?
Ma'am, no. Like we understood.
Or you are clear with it? I hope you
Clear.
are clear. It's just that I thought I have written it as capital Y, but it is small y. Okay. So then when we will write, we will fill in the boxes now. The phenotypic ratio will turn out to be 9 is to 3 is to 3 is to 1. As in 9 seeds would be round yellow in color. Okay. The 3 seeds would be, come on, tell me which color. Come on, quickly tell me.
Ma'am, wrinkled yellow in color.
Wrinkled, yellow. And what about next 3?
Wrinkled green in color.
Round green in color.
Sorry. Yeah, I mean like around
Okay.
green in color, and the
And
one
only
next to
one,
it
that
is
is the last. Small r, small y, small y. It is for wrinkled green. Okay. Okay, come on. Tell me if the dihybrid cross is clear to you. Let me show you the last slide. I know the time is up and you must be waiting for me to end the class. But we are almost done. See this quickly. See this slide. Okay. See this slide. So this is the dihybrid cross that we have got, and this is the phenotypic ratio 9 is to 3 is to 3 is to 1. This you have to learn. Okay. So here in the F2 generation, only one law of segregation. What does the meaning of what is the meaning of law of segregation? What is the meaning of segregation? I'll just quickly cover it. Okay. Segregation means separation. Separation. Suppose you have got a pencil box, okay, a pouch in which you have kept pencils, eraser, sketch pens. You have kept a lot of things in that single pouch. Now, if I ask you to separate all the stuff, how are you going to do it? You will be taking out the pencils and keeping it in a separate place. You will take out the sketch pens, keep it separate. You will take out the erasers, you will keep it separate. Why I'm giving you this example? To tell you that you are actually segregating or separating the things which are there in your pouch, right? You have separated and segregated the sketch pens, pencils, and erasers. Similarly, there is the segregation of gametes also which happens. Law of segregation means this only. What does this law states? Okay, so the law of segregation states that when a pair of factor for a character brought together in a hybrid, They will separate during the formation of gametes. And this is what we have been doing till now. Suppose there was a parent, there was a hybrid condition as capital T, small t. How you were making the gametes? You were separating it. Like you separated this and you separated this. So this is what? Law of segregation. Is that clear?
Yes, ma'am.
You have just you have just separated the pair of characters during gamete formation. Okay, so this is the hybrid condition. That means one is capital T, one is small t. So when the gamete formation has to take place, how we did it? We separated them, one capital T, one small t. So this happens for the formation of gamete, and this is known as law of segregation. Just give me 1 minute. I'll quickly cover law of independent assortment as well. It's very simple because we have done the cross. It will be clear to us. In this, okay, who will tell me that what are the parents in the dihybrid cross? Mendel took what kind of parents? Quickly tell me.
Round and yellow, wrinkled and green.
Correct. So he took these two parents, but out, out of his surprise, in this dihybrid cross, what new combinations did he get? Were all the seeds round yellow? Were all the seeds wrinkled green as like parents? Or there were some new combinations as well?
F1, all of them were round and yellow. F2, they were both.
Yeah. In F2, there were some new combinations seen by Mendel. And what were those new combinations? Quickly
Round
tell
and
me.
yellow, round and yellow, wrinkled and yellow, wrinkled and green, round and green.
No, 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 no. Shivansh, new combination that was different from the parent.
Oh,
Parent
wrinkled
was,
and yellow and round and green.
yeah, wrinkled and yellow because it is not like the parent. Okay, and round and green, it is also not like the parent. These were the new combinations, right? But how do these new combination formed? Now here comes the law of independent assortment. What does that mean? Just 2 minutes. Okay, just 2 more minutes. Law of independent assortment. See this very carefully. It states that factors for different pair, factors means genes. Different pairs of contrasting character in a hybrid, they are distributed independently during gamete formation. Suppose हमें समझ नहीं आया ये क्या लिखा है. No worries. Come to the cross. See this. Can you see this gamete formation, children? Here.
Yes, ma'am.
Okay. In the F1 generation, what were the gamete? The gametes were capital R, capital Y. Small r, small y. Right? As in, round was always with yellow and wrinkled was always with green. Yes or no? Just talk
Yes.
about the F1 generation. Round was always with yellow. Wrinkled was always with green. Right? This is the story of F1 generation. But what happened in F2 generation? Round got a new friend. What new friend? What new friend? Round got combined with a new friend. What new friend?
Ma'am, wrinkled.
No, no, no. See the gametes. See the gametes. See these four gametes. Tell me that round apart from yellow got a new friend. Who is that new friend? Not un You're not able to understand what I'm saying? Okay, let me come to this. See this. Can you see the Punnett square of F2 generation?
Yes, ma'am.
Children, see. This is round yellow. Yes or no?
Yes.
This capital R has got a new friend also at the time of gamete formation. Who is that new friend? Capital R out of these three combined with what? Green? Round green?
Yeah.
Yes or no? You
Yes,
got
ma'am.
to see round green seeds also, na? In F2
Yes.
generation. In F1, round was always with yellow. He used to stay with yellow, for example. Imagine. Assume that round used to always stay with yellow. But in F2, we saw a new combination. What? That this round got with green also. Okay. Similarly, in F1 generation, wrinkled was always with green. But in F2, what new thing we saw? We saw that wrinkled got with yellow also. We got to see wrinkled yellow seeds also, na, which we didn't see in the F1 generation. We got to see round green seeds in F2 generation, which we didn't see in the F1 generation. That means what? We saw new combinations of in F2 generation. That means round is not completely dependent on yellow. No, he is not dependent on yellow. He can independently assort, as in it's not dependent on yellow. It's independent because it can go with green as well. This is law of independent assortment. That means the gametes, they are not dependent on each other just like F1 generation. In F2 generation, they are independent. That means they are not going to stick with yellow always. R is not going to stick with yellow always. Okay, it went with green as well. That is why you got to see round green seeds in the F2 generation. Same with wrinkled seeds. In F1 generation, wrinkled used to always stay with green color. But in F2 generation, what we saw? The wrinkled went with yellow color also. That is why the seed became wrinkled yellow. That means wrinkled, that means it's independent. These are independent. These are independently assorting. They are independently distributing. That is why you get to see the new combination. And this is the law of independent assortment. Nobody is dependent on each other. They are independent. That is why they are moving out and forming new combination of seeds that you didn't see in the F1 generation. Got it or not?
Yes, ma'am.
Everybody. Okay, how many of you want me to explain law of independent assortment again? Don't worry, I'm not going to explain it today because time is up. I will explain in the next class. But how many of you still want me to explain this law of independent assortment again? Quickly tell me now.
Ma'am, you can explain this again.
Huh? You want me to explain again in the next class?